So here I have my bacteria. It's already been streaked onto here, incubated, and is ready to go for me to perform a bacterial smear. So with a bacterial smear, start with a clean microscope slide. And you have a couple options in this first step as far as which kind of tool to use. This is a wax marker. You might also know it as a China marker, and this is a Sharpie. Depending upon what kind of staining procedures you're doing, if you use a Sharpie in any sort of decolorizing step, that Sharpie can actually get erased and bleed over and contaminate. The colors can contaminate your sample. A wax marker, though, can be kind of hard to use and hard to draw on this slide. For my students, there's a couple different things, depending upon how comfortable they are, that I might recommend they do. I might ask them to write their initials along this top edge so that they know that this part is the top of their slide. And that will come into play a little bit later. And then drawing a circle along the middle third of your slide with this wax marker. So here you can see that there is a circle that I've drawn. And this is actually going to let me know where I'm going to smear my bacteria and help me keep track of it. So I won't be using the Sharpie for this one, but I did want to show you it is an option, especially with if you're just going to do a simple stain. But if you're doing a more complicated stain, you may have problems with that Sharpie being erased between all the rinsing steps and the decolorizing steps you see in more complicated procedures. So now that I have my circle, the next thing I need is to actually add water to my slide. So this is distilled water. I only need a drop, and actually a really tiny amount. So there's this fun technique that was taught to me where you can help gauge how much water you need on your slide using the tip of an inoculating loop. So this requires me to first sterilize this. So I'm actually going to use a hooded igniter to light my flame. And so I'm going to keep the loop end down to sterilize this inoculating loop. I'm going to start at the base, wait for it to get red hot before I move on to the next section. All right. So this loop has been sterilized. And then this next part, it's a little bit tricky and probably even trickier to capture on camera. But I'm going to attempt to trap a water droplet into that loop. So this water droplet, it's pretty big. I'll bring it a little bit closer to you guys. Maybe you can make it out. There you go. That water droplet's pretty big. So I'm actually going to want to tap to break that water droplet. And then I'll show it to you again one more time. See if I can get in focus. There you go. So there's a thinner film of water trapped in this inoculating loop. And I'm going to use that thinner film of water to tap it on and then add it to my microscope slide. So that's not a ton of water. And then this is a part that requires me to act a bit quicker, but I'll set this slide down. I would then sterilize this loop another time, wait for all sections to get red hot. All right, I would then wait for this to cool. I can also touch it to a clean part of that agar. Now I don't want to grab too much because then my sample will be crowded and all the microbes will be overlapping each other. So really, because this loop is sterile, all you really need to do is touch the surface to a colony. There's no need to do any sort of aggressive scraping of the whole plate. And even if you do that, you risk breaking the agar or grabbing way too much bacteria. So you need to focus on a small section, ideally an isolated colony. Tap this sterile loop to that colony, and you don't want to see like that whole loop filled with bacteria. In fact, you may not see anything at all, and that's perfectly fine. But this isn't a spoon. You're not trying to scoop anything out or coat the entire surface so much that it's visible, because then again, you'll have way too much bacteria. But just imagine this inoculating loop is divided into like 10 sections. For instance, you don't want to see bacteria all over all 10 sections, maybe just over 1 or 2 sections out of 10 on this inoculating loop. That's just the first time when you're eyeballing it and you haven't done it before. If you just want to be awfully sure you have microbes on this loop, but imagine that you are I almost describe it as like squeegeeing. This has the consistency of similar to Jello, and the microbes on top are similar to whipped cream, and so you have to be gentle. In reality, all you actually have to do is just tap it. And even if you don't see anything, as long as you're not twisting this loop around, then you should successfully transfer microbes. So I'm not actually going to unwrap this for this demonstration, but imagine that I did add microbes to this loop. I would then stir those microbes into this water, making sure I'm staying on my circle, staying inside my circle. All right, and then after that, I will leave this slide up here to dry. I'll sterilize my inoculating loop again. So once that dries, you won't see any water along the surface. If I had added actual bacteria onto it, you may see a thin film after this dries. So depending upon your slide setup, you may have access to a slide warmer, or you can gently tap your slide to the surface of that warmer to help that water droplet disappear a little bit quicker, so you all don't have to wait as this slide dries. I've gotten another slide, but in reality, as long as you've used a really thin film of water trapped along this loop, it will only take a couple minutes to dry. But so you guys don't have to wait for that, I've gotten a clothespin. I'm going to use this to clamp my microscope slide in that has my bacteria, and I need to heat fix it. If I forget to heat fix it, once I do subsequent steps like like a simple stain, it's going to erase everything on this microscope slide. So for heat fixing, all you need to do is take your bacteria and pass it over the flame about 3 times, making sure that center is going over the flame. So this is the bottom of the slide. My bacteria were added to the top of the slide. So once, twice, and 3 times, and that is it. You might want to wait a second or two for your slide to cool, but after that, that's actually the final step of the bacterial smear itself.